Hi, in this video, we will share with you guys about downstream processing, focusing on microbial product separation. What is downstream processing? Downstream processing is where the cell mass or products from the upstream are processed to meet purity and quality requirements. There are several typical operations used in microbial product separation, which are Froth flotation, sedimentation, centrifugation, precipitation, and filtration. We're going to focus on two operations, which are Froth flotation and centrifugation. What is Froth flotation? Froth flotation is a method to separate hydrophobic from hydrophilic materials. It is also a mineral separation process that takes place in a slurry of water and minerals. Froth flotation can only be used for relatively fine particles. If they are too large, the particle-to-bubble addition would be less than the particle weight, causing the bubble to lose its load. To render the hydrophobic or hydrophilic particles and to facilitate bubble attachment, flotation reagents must be added. There are three types of reagents: the collectors, the frothers, and the regulators. Non-ionic, anionic, and cationic collectors are the three types of collectors available. Simple hydrocarbon oils are used as non-ionic collectors. The polar component of anionic and cationic collectors selectively binds to the mineral surfaces, while the non-polar component projects out into the solution, making the surface hydrophobic. For frothers, this reagent produces stable bubbles for hydrophobic particles to bind. It can also act as collectors. Pine oil and MIBC are examples of frothers. Regulators, often known as modifiers, are widely used in flotation to change the collector's action by increasing or decreasing the collector's water repellent effect on mineral surface. Activators, depressants, and pH modifiers are all types of regulators.
Method of froth flotation. This diagram illustrates the components that involve in froth flotation process, which include tank, agitator, sulfide ore, water, pine oil, and compressed air. In order to give a clear view, this illustration will show how it actually works. In this process, the tank was filled with water and pine oil as a forming agent. This involved a wettability principle in order to get the ore separation. Then the powdered ore is added and stirred vigorously with agitator and by blowing the compressed air. This results in the formation of the froth or foam. After froth are formed and rises to the surface, the impurities or GAM particles are left or settle down in the tank. This is the view on how the bubble form. Some industries will add a depressant, which is sodium cyanide, to the mixture in order to decrease the association of GAM particles with the froth. Lastly, the froth is removed, collected, and transferred to the other container for further processing.
Since froth flotation is a process that can selectively separate materials based upon whether they are water-repelling, hydrophobic, or have an affinity for water, hydrophilic, this technique is commonly used in various industries, including mining industry. The particles of interest are physically separated from a liquid phase based upon their hydrophobicity. The hydrophobic particles with the air bubbles attached are carried to the surface, thereby forming a froth which can be removed, while hydrophilic materials stay in the liquid phase. Froth flotation has also a significant place in the waste management and waste recycling or recovery industry. Example of industrial applications of flotation for environmental problem solutions includes pulp and paper industry, textile industry, water purification, industrial waste and sewage, food industry, and plastic recycling industry. It is believed that flotation will find new potential application areas and gain more importance in waste management and recycling or recovery industries in the near future.
Other than froth flotation, centrifugation can be applied for harvesting products after fermentation. This process requires a machine called a centrifuge. A centrifuge is a device used for separating particles from liquids by gravitation forces. Denser components will migrate away from the axis of the centrifuge and less dense components migrate towards the axis. These are the factors that will affect centrifugation. First, the density of samples and solution. The larger the density of the particles allow particles to separate faster from the mixture. Next, the temperature and viscosity. The temperature within a rotating centrifuge rises as the energy inside it increases. Temperature has an effect on the viability of some analytes, and therefore, refrigeration helps to keep the sample stable. Furthermore, the effectiveness of a pump can be greatly reduced as viscosity rises. Lastly, the distance of particles displacement and rotation speed also affect centrifugation. There are many applications of centrifugation. This process can be used to separate two miscible substances, purify mammalian cells, fractionate subcellular organelles and membrane vesicle, separate fats from milk to produce skim milk, and many more. Centrifugation is applied both in the laboratory and on an industrial scale. The centrifuge used in the laboratory usually looks like this. Here is a clear example of how the solutions react during centrifugation. In big factories, centrifugation is part of their downstream processing. It is used to recover cells and other valuable pharmaceutical reactants and products used for sewage processing and isolate valuable synthetic materials. An industrial centrifuge looks way different than the ones used in the lab. Moving forward, different centrifuge works for different purposes. However, their basic working principle is applicable for all applications.
Bioprocess Technology, Industrial Centrifuge. What is industrial centrifuge? Industrial centrifuge is a machine used to separate and collect the product of interest such as enzyme, antibody or mammalian cells from the culture media in the downstream process. How does an industrial centrifuge works? After the fermentation process, the broth tank is connected to the T-stack centrifuge. The tank is pressurized to drive the broth into the centrifuge. The centrifugal force causes the solids to move and collect at the side of the bowl. The liquid flows through and out of the centrifuge. As more and more broth enter the centrifuge bowl, the clarified liquid is moved to the exit at the top of the bowl. Meanwhile, the solid that contains the desired product will continue to collect at the bottom of the bowl. There are three types of thermoscientific centrifuge. The first one is thermoscientific batch centrifuge. The second one is thermoscientific batch centrifuge or zonal continuous centrifuge. And the last one is thermoscientific zonal continuous centrifuge. That's all for industrial centrifuge. Thank you.